Greetings, class. I'm Mr. Morales with today's science lesson on mitosis. Mitosis. We're going to learn about the steps of mitosis. Mitosis is the process cells go through to reproduce, and it occurs in multicellular organisms such as the man, the plant, the trees, the bugs, and all of the things, all of the living things that you see in this picture. Before we get started, let's review the examples of multicellular organisms. Let's talk about eukaryotes. Eukaryotes are usually multicellular organisms that have a true nucleus and membrane-bound organelles. They are more complex than single-celled organisms, and they have cells with specialized roles. Think about brain cells, skin cells, heart cells, muscle cells. Those are cells found in eukaryotes, multicellular eukaryotes we're talking about. Examples of eukaryotes include animals, plants, fungi, mushrooms, which you see in the picture, and protists, microorganisms that can be single-celled or multicellular with membrane-bound organelles and a proper nucleus. In order to be a eukaryote, your organism must have a nucleus. Inside the nucleus is where the DNA is located. And remember, eukaryotes must have membrane-bound organelles. Eukaryotes replicate their cells for growth and development. You'll learn more about this process over the course of this lesson. By the end of today's lesson, you should be able to do the following. Describe the steps of mitosis, the individual steps of mitosis. Explain the importance of mitosis to living organisms. Finally, you'll be able to analyze new technologies and experiments and see how they affect previous scientific explanations. What is the process responsible for the replication of cells for growth and development? You began by addressing this question in the warm-up when you learned eukaryotes replicate cells for growth and development. You'll now answer the question specifically as you learn about the process called mitosis. Let's talk about the cell cycle. The cell cycle incorporates two stages, interphase and mitosis. Interphase is this large section you see in green. An interphase can be further divided into smaller sections, which we'll discuss now. G1, seen here, stands for gap 1. Gap 1, or growth phase. During interphase, the cell starts to grow. The cell's DNA is getting prepared to be replicated or duplicated. During S is synthesis, where the DNA is actually duplicated, meaning an exact copy of the DNA is being made. And G2 represents growth, or once again, gap phase 2. The growth is needed for the cell to produce more cellular material so that it will eventually be able to divide into two cells that look like the original cell. So the cell has to grow significantly from its original size. Chromosomes are duplicated during interphase, allowing there to be two identical copies of DNA. You will not be able to tell the offspring cell from the parent cell after mitosis is complete. Interphase, remember, is not part of mitosis. Rather, it comes immediately before mitosis, but is extremely important in the process. It's extremely important because it's the process the cell goes through right before division. Now let's talk about mitosis. Mitosis is the process of cell division by which exact copies of chromosomes are divided to create two daughter cells, each with a complete set of identical chromosomes. Now, let's move on to the stages of mitosis. Prophase. Prophase is the very first stage in mitosis in which the chromatin in the cell coils into chromosomes that now can be seen as you look into the cell's nucleus. The next stage is metaphase. Metaphase is the second phase of mitosis in which the chromosomes that were doubled become attached to the spindle fibers by their centromeres. Centromeres are in the center. The next phase is anaphase, the third phase of mitosis, where the centromeres are now split apart and the sister chromatids are now pulled to opposite poles of the cells. The chromatids contain the same information about the cell that is dividing. After anaphase comes telophase, the final stage of mitosis in which the sister chromatids are now completely separated, and they contain, remember, identical copies of DNA. Also in telophase, you see that the nuclear envelope has reappeared, and you start to see what are becoming two different nuclei. There's a key way to remember these stages of mitosis. We use an abbreviation that's called PMAT. Letters P, M, A, T. Remember PMAT. P for prophase, M for metaphase, A for anaphase, and T for telophase. These stages will be seen over and over again. I want to review them one more time, though. Prophase, once again, first phase of mitosis, in which the chromatin in the cells coil into chromosomes that can be seen. Metaphase, the second phase of mitosis, in which the chromosomes that were doubled become attached to the spindle fibers by their centromeres. Remember, the third phase is anaphase, the A in PMAT. This is where the centromeres split apart and sister chromatids are pulled to opposite poles of the cell. And of course, the last phase being telophase, the final stage where the sister chromatids are separated, and these chromatids contain identical copies of DNA. They're now on opposite sides of the cell that will soon divide.
Let's discuss each phase in detail of the steps of mitosis. Let's start with prophase. Well, prophase is the first and longest phase of mitosis. The first phase of mitosis, in which the chromatin in the cell coils into chromosomes that can be seen. So chromatin is the DNA that's all spread out so the cell can use it, and when it condenses into chromosomes, it's actually condensed into the interior of the cell. Now, during prophase, the nucleus, nucleolus, and nuclear membrane begin to disappear as they dissolve to prepare the cell for division and allow movement of chromosomes. So chromosomes become visible as the chromatin as the cell begins to coil together. And you can see them right there. A chromosome is actually, and that's what we're looking at here, a chromosome is actually a cell structure made of DNA that gives the master instructions for the cell. Sister chromatids are located on either side of the chromosome and contain identical DNA, which is duplicated during interphase. Now, sister chromatids are exact halves of the chromosome from the parent cell that are connected at the centromere. You see that right there. After cell duplication, but before cell division. Now, sister chromatids held together by the centromere, and that's the structure that holds the sister chromatids together. Now, centrioles migrate towards the opposite ends of the cell. So you can see these moving towards opposite ends of the cell. Centrioles are actually a pair of cylindrical structures found near the nucleus in animal cells, composed of microtubules that are copied during interphase and move to opposite ends of the cell during prophase of cell division. Spindles, and you can see these beginning to form right here. Now, metaphase is the second phase of mitosis. It's where the chromosomes that were doubled become attached to the spindle fiber by their centromeres. It's a short phase. The doubled chromosomes attach to those spindle fibers, and the spindle fibers push and pull the chromosomes, and the chromosomes are lined up across the center of the spindle. Each chromosome has their own spindle fiber. Now, one spindle fiber of a sister chromatid is connected to one end of the cell. The other chromatid spindle fiber is connected to the other end of the cell. So they're connected, one at this end and the other one down at that end. This is important because as the cell divides, each half of the cell will receive identical copies of DNA from the chromosomes. Anaphase is the third phase of mitosis, and it begins when the sister chromatids separate. Okay, when those centromeres are split apart and the sister chromatids are pulled to opposite ends of the cell, this is anaphase. Now, during anaphase, the centromeres split apart and chromatid pairs move towards either end of the cell as those spindle fibers shorten. Scientists are not sure how the chromatids move, but they believe it has to do with that spindle shortening. Telophase is the final stage of mitosis, where sister chromatids are separated and contain identical copies of DNA. The chromatids then reach the opposite ends of the cell. Many of the changes that took place during prophase are reversed because the new cells are preparing for an independent existence, and mitosis is done at this step. The cell is still connected, and there is one cell with two identical copies of DNA. The next slide, well, it's going to explain how the cell separates. The spindle fibers break down, nucleus is formed around each set of chromosomes, the cell membrane begins to separate into two different nuclei. The chromosomes unwind so they can begin the metabolic activities of the new cells. So, cytokinesis is the process that divides the cytoplasm and plasma membrane of a cell. It results in two identical cells containing their own DNA, nucleus, nuclear membrane, and plasma membrane. So, cytokinesis is not a part of the process of mitosis, but it does start during anaphase and completes during telophase. It divides the cytoplasm, it results in two cells, and it is different processes for plants and animal cells. There are two cells for the plant cell and two cells for the animal cell that are created as a result of mitosis. In a plant cell like this, you have a cell plate that begins in the middle and grows outward towards the cell wall to form two separate cells. In animal cells, the plasma membrane pinches in here until it grows together and meets in the middle, and it allows the cell to divide into two cells with identical DNA.
What is the process responsible for the replication of cells for growth and development? Eukaryotic cells replicate for growth and development through a process called mitosis. You'll learn that the steps of mitosis are prophase, metaphase, anaphase, and telophase. Remember that abbreviation, PMAT. The end result of mitosis is two identical cells. But have you ever wondered why a cell needs to reproduce at all? Why does it need to divide? Next, we're going to look at the importance of mitosis to living organisms. Living cells depend on mitosis for life. Mitosis provides organisms with growth. All cells are divided and duplicated, and new cells are formed. This allows organisms to continue to grow. When a baby is born, it's not the same size as an adult. Look at the small horse and the adult horse alongside of it. The DNA in the cells of the baby is duplicated, and then the cells are divided. As these cells divide, the new cells help in the growth of the baby from childhood, babyhood, to adulthood. Their body cells, skin cells, heart cells, brain cells, bone cells, and etc. all continue to reproduce for the growth of the baby. It's the same way with plants. They start out very small and grow as cells are duplicated and divided during mitosis. Organisms also depend on mitosis for repair. The duplication of cells during mitosis allows new cells to be formed that replace old or damaged cells. The duplication of cells allows new cells to replace cells that can be damaged in the environment or cells that die from old age. Without mitosis, old cells would never be replaced because new cells would not be formed to replace them. Reproduction also occurs, creating new cells. Look at the plants and the cells below. Liver cells reproduce only liver cells. Lung cells produce only lung cells. Cardiac cells produce only cardiac cells. Mitosis allows each type of cell to produce its own type of cell for the proper functioning of an organism. Your body cells go through mitosis continuously for growth, repair, and reproduction. Ultimately, the purpose of mitosis is to allow organisms to grow, repair, and reproduce new cells in order to survive. Now, let's analyze how new technologies and experiments have affected previous explanation. Tissue cultures can be seen in many laboratories today. Original body cells are removed from the organism and placed in a sterile medium to be studied. While observing the cells in the medium, cells reproduce. They undergo 20 to 50 mitotic divisions. The cells only grow one layer thick, and then they stop producing. When the cells are touching, they do not reproduce. This is called contact inhibition. If the cells are no longer touching because of a death or damage to a cell, cells will reproduce to fill the empty space, but they will not grow or reproduce any further once the empty space is filled. Now, there can be changes in situations like this. A mistake in mitosis creates cells with different DNA from the original cell. New cells that grow and reproduce without limit are called cancer cells. Understanding how the mitosis of normal cells changes and works allows scientists to use tissue cultures to study cancer cells. When cancer cells are grown in a sterile medium, they reproduce rapidly and form multiple layers. There is not a limit to the number of generations they can reproduce. Remember, healthy cells stop after 20 to 50 mitotic divisions. Cancer cells continue to grow without stopping. This is because cancer cells lack contact inhibition to tell them when to stop dividing. Previous to using tissue cultures, scientists were unclear how cancer cells were able to divide so rapidly. Let's look more about tissue cultures and talk about contact inhibition. The study of mitosis in the past has led to the study of tissue cultures. Through research and experimentation, scientists discovered cancer cells lacked contact inhibition regulators. They also discovered that they could study cancer cells the same way that they studied healthy cells. Using cancer cells in a culture allows doctors to study which medications will best fight cancer cells in a cancer patient. In the past, a patient would be subject to one toxic medication after another in hopes of fighting the cancer cells. Using tissue cultures allows doctors to treat patients that have cancer more effectively without experimenting on the patient. What happens is a biopsy is taken. A culture of cancer cells is grown in a petri dish after a biopsy of the cancer is removed from the patient. This cancer grows out of control. Unlike the healthy cells, which produce only 20 to 50 mitotic divisions, cancer cells grow out of control in the petri dish. Scientists can then inject the cancer cells with medication to study which medicines will work best to stop the cancer cells from dividing, growing, and multiplying. When they find a medicine that is working to fight the reproduction of cancer cells, they can then use that same medicine on a patient, using the same patient that the biopsy was taken from. This saves the patient from receiving experimental medicines that are toxic and allows them only to receive the medication that has worked on the biopsy cells that the doctor had taken from them previously. It's very promising in the research of how to work and how to defeat cancer cells.
Today's topic is the cell cycle and mitosis. But before we get into the cell cycle and mitosis, we want to make sure and talk about why these words are important and how they relate to real life. So if I say the word cancer, this word probably means something to you. Either you know somebody who's been diagnosed with cancer or you've heard about it on the news. There's many different types of cancers, but one thing they all have in common is that they are caused by uncontrolled cell growth. That means somebody's own cells grow out of control. Now, that doesn't mean that there aren't other contributing factors. For example, for some cancers, having genetic links, meaning it runs in the family, can make somebody more likely to develop it, or having excessive exposure to UV light. For some cancers. Those are risk factors that can increase your risk, but the major cause of cancer is uncontrolled cell growth. That also means it's something that is not contagious, and it's something that anyone can develop. You have to realize that all the cells in your body are very, very busy, and they're either in one of two different phases. They are either in a phase called interphase, and during interphase, they are just growing, they're replicating their DNA, they're doing their cell functions, or they are in mitosis. Mitosis is a fancy word that means that they are dividing, they are making new cells. Well, if you think about the cell cycle, whether they're in interphase or mitosis, it's kind of like a pie graph. They're spending about 90% of their time in interphase. Most of the time, cells are not dividing. Most of the time, cells are in interphase. They're growing, they're doing their daily cell functions, and they're replicating their DNA. Most of the time, cells are not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells, they do mitosis frequently, which is why your hair can grow up to half an inch every month. But other cells, like, for example, your brain cells, your neurons, some of them actually do not perform mitosis, which is why if you have some type of major injury to the spinal cord or to the brain, sometimes those do not heal very well because those cells, they do not replicate. So mitosis is very important in order to generate new cells, which can help with growth. And also, if you damage your cells, if you're walking down the street and you have some kind of accident and you skin your knee, well, you're going to need to repair those damaged cells. You're going to need to be able to do mitosis to make those new cells. The thing is, you don't want your cells to always be doing mitosis. Because if they're always doing mitosis, then they're always dividing, and then they're not carrying out their actual function. And that's a problem. That's where checkpoints come in handy. See, along the cell cycle, there are checkpoints to check that the cell is growing correctly and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. And what happens if the cell fails its checkpoint? The cell does something called apoptosis, which basically means the cell destroys itself. It self-destructs. This might seem kind of harsh, but actually it's really important because it ensures that you don't have these cells that are messed up continue on and divide. You don't want damaged cells to continue on and divide because they might not do what they're supposed to. They might start to grow out of control. They might not be regulated correctly. That's where cancer cells come in. See, cancer cells, as we had said, are your body's own cells that are growing out of control. So they're not following the rules of these checkpoints. They're flying past the checkpoints, going through the cell cycle, and frequently doing mitosis, which means that they're dividing very, very often. And so by doing that, they can grow into these things called tumors, which are really a mass of cancer cells. So we're not really sure what causes them to escape these checkpoints. There's a lot of risk factors that that a person can have that can increase their risk of this happening. But when these cells get past these checkpoints and grow out of control, they become a problem because many times they can travel through the bloodstream. They start growing out of control. The body can't identify them and destroy them with the immune system. So this becomes an issue. And And usually, one of the treatments that somebody who has cancer will undergo is chemotherapy. And this works by targeting cells that go through the cell cycle quickly and hit mitosis quickly. And that's one reason why somebody often will lose their hair when they're on chemotherapy, because they're on this medication that's targeting fast-growing cells. And your hair follicle cells are also fast-growing cells. Cancer cells have some other factors that make them difficult to treat. One is that cancers have the ability to secrete their own growth hormone. This is very bad because growth hormone, in a sense, think of it as a hormone that says, "Feed me." And what happens is the blood vessels will divert over to those cancer cells and supply the cancer cells with nutrients. This is not good because this means the cancer cells will have the nutrients they need to keep growing out of control. It also can take nutrients away from nearby healthy cells. There's some medications that are being researched that maybe could stop the growth hormone from cancer cells. And in a sense, that would be great because the cancer cells would not be able to get the nutrients to grow out of control anymore. Of course, you have to make sure that the medication is targeting just the cancer cells and that you're not affecting the growth hormone of nearby healthy cells. You wouldn't want that. So it's kind of a tricky situation. So let's recap what we talked about today. We talked about the cell cycle, how cells are more frequently in interphase than they are mitosis. Interphase is when cells are replicating their DNA or they're growing, carrying out cell processes. In mitosis, the cells are actually dividing. We talked about how during this cell cycle, there's checkpoints that regulate the cell and keep the cell from being able to continue on if it's damaged. Unless we're talking about cancer cells, which are cells that have escaped the checkpoints and are growing out of control. Be sure to check out our clip on mitosis. This process is really fascinating, but it's also a really important one in order to understand how your body cells work. That's it for the